Hello, everyone, and thank you for joining us virtually today. My name is Carl Radosevich, and I'm coming from PHC Corporation of North America here in Chicagoland. We operate under the PHCbi brand of equipment, and today I'm going to discuss several technology innovations in cell sorting, 3D imaging, and oxygen control, all with important impacts on the stem cell workflow. So thanks for being with us today. A quick company introduction. At PHCbi, we are a Japanese-based life science equipment manufacturer. In short, we're experts on the technology for environmental cell culture control. And as you can see from this visual timeline, we have a rich 55-year history as a life science equipment manufacturer. Our innovation is of the Sanyo and Panasonic legacy. So if you see cold storage and incubator units with Sanyo or Panasonic logos, those are our proud legacy of products. Although our name has changed, our innovation and technology run deep and is proven in thousands of research labs worldwide. And throughout this process, we worked very closely with top innovators and pharmaceutical organizations across the world to evolve with the ever-changing needs of science. This is especially important in the age of live and biologics-based medicine. Leading into today's technology discussion, our PHC company is motivated to stay on the cutting edge in new product development, both organically and through strategic partnerships globally. Today, we'll share information on our technologies as they pertain to advancements in cell culture, specifically surrounding 3D spheroid and stem cell studies. Most recently, we've extended our technology solutions portfolio with the addition of a microfluidic cell sorter through partnership with On-Chip Biotechnologies, and a variety of 3D live cell imaging platforms through Screen Life Sciences. Also, we have unique 3D cell culture plates called PrimeSurface. Together, our portfolio solutions provide a unique equipment solutions for the regenerative and stem cell research community, from culturing and preservation through sorting and analysis. Thanks again for joining us. So without further ado, I will dive into the cell sorting equipment first. I'd like to take the opportunity to introduce a new collaboration between PHC and On-Chip Biotechnologies. At On-Chip, we have two systems that would come in handy for stem cell and spheroid research. The On-Chip Sort is our microfluidic cell sorter, and the On-Chip SPIS, or SPICE, is our single cell or spheroid dispenser. But let's go ahead and start with our cell sorter. First, let's revisit some general flow cytometry principles. Here's a schematic that describes how what we call the conventional cell sorter works. Conventional cell sorters use high pressure to drive samples through narrow tubes, and the cells get injected out from this nozzle. As they come out of the nozzle, they get encapsulated inside liquid droplets, and those droplets containing the target cells are then charged to electrostatically collect the cells into a container. Those steps induce stress to the cells that are being sorted, and those cell damages can be expressed in so many different ways. This means your sorted cells would not behave the way you wish them to, as shown here. And so we've come up with a completely new sorting method using a microfluidic chip. Cells travel through a narrow microfluidic channel while being detected by lasers. Cells that we do not want simply travel straight through the channel, but when we detect the cell of interest, we generate a gentle liquid pulse to push the cell across to the side. This way we eliminate all the damaging steps involved in the conventional sorting technique. It means that we can handle a much wider variety of sample types that are particularly fragile, and that is our main topic of the day, which will focus on spheroids. When you're doing some assays using spheroids, you really need to use spheroids of similar sizes for the assays to really be reliable and have reliable data. However, spheroid sorting has been a challenge for so many research due to the size. Standard cell sorting systems can only sort particles usually up to about 45 microns at best. This is due to the nozzle that they're using, and the spheroids that manage to get through the nozzle usually end up breaking into multiple pieces. On-Chip, on the other hand, can sort spheroids up to 140 microns. We're also working closely to reach that 300 micron mark. So in this example that is shown on the slide, we've decided to sort spheroids by size from a culture with a variety of spheroid sizes. The size varied from a single cell level up to about 200 microns, but we've successfully isolated those spheroids of a size around 100 microns. Looking at some stem cell workflows, here's a technique utilized by Dr. Chumba's group at Kyoto University. In the field of regenerative medicine involving ES or iPS cells, differentiated cells are often cultured to tissues and implanted into patients. But there's a problem that some remaining undifferentiated cells in the implanted tissues may cause tumors. Therefore, a complete removal of undifferentiated cells prior to implantation is important. For this purpose, we've developed a novel technology called multi-step negative sorting to completely remove undifferentiated cells. This technology also provides an absolute quantification of iPS cells as a quality control of cells for transplantation. Conventional cell sorters are typically used for purification of cells, but they're really not suitable for use in regenerative medicine because those cells tend to be damaged during sorting. To overcome this inherent problem, our microfluidic chip-based damage-free sorter uses a disposable and sterile microfluidic chip, again, as the core of its technology. Target cells are isolated by pulse flow and created by pneumatic actuation. The figure on the right shows the method of multi-step negative sorting on the on-chip sort for the complete removal of undifferentiated cells. This method works in the opposite way to conventional sorting methods. Samples loaded on the sample reservoir of the chip, the undifferentiated cells or the unwanted non-target cells are removed by formation of pulse flows into the collection reservoir, while the differentiated cells or the target cells flow into the waste reservoir located downstream of the chip, where those cells are retained rather than being discarded like the conventional cell sorters. Differentiated cells are recovered and reloaded on the sample reservoir to be sorted again for any further purification. This process is repeated until all the undifferentiated cells are indeed removed. The method only requires sorting speed that corresponds to the detection rate of the rare cells. When a percentage of the undifferentiated cells is 1% of the whole population, roughly, then this negative selection is 100 times faster than collecting differentiated cells using positive collection. So just like any other cell sorter, we can detect up to 6 fluorescent signals off 3 different colored lasers. This allows for detection and sorting of spheroids based on fluorescence also. And not only spheroids, but many other types of cell cluster samples can be manipulated and observed on the on-chip sort. Shown here we have some tissue fragments from brain, organoid of the intestinal crypt, worm eggs, long cells like cardiomyocytes, and living organisms like protozoa. All of these cell types can be sorted on the on-chip sort with no damage to the cell samples at all. So I assume that you know we can handle large particles in different types of cells by now, but our cell sorter itself is actually much smaller than the conventional sorting technologies. And for this reason, we can place our sorter inside a biological safety cabinet to handle spheroids, stem cells, or any other regenerative type of tissues in a very sterile condition. Furthermore, our microfluidic chip is sterilized itself, so you can be assured that all the samples are handled in a perfectly sterile and clean environment. Not only that, since our microfluidic chip is disposable, there will be no sample carryover from the previous sample either. This is actually particularly important when handling clinical samples, as I'm sure many of you are. So now we'll move on to our next instrument, our single cell dispenser, the on-chip SPIS. I assume some of you have tried placing single spheroids into well plates, but this is no easy task. One way to do this manually is to dispense single spheroids by limiting dilution. However, this will only give you about 1/3 of the wells being single, and the rest will either be empty or have more than one, creating a lot of inconsistencies as shown on the left. Our single cell dispenser, the on-chip SPIS, ensures that almost all the wells would have a single spheroid. So this is how it works. We use a very thin pipette tip to aspirate the sample and use a camera actually to detect how many spheroids are inside that tip. Only when we have one spheroid inside the tip, we go and dispense that into the well. We can ensure that all the wells would have a single spheroid. The dispenser allows for dispensing of spheroids up to 200 microns in size. The plate can either be a 96 or 384 well plate, U-shaped, V-shaped, or flat bottom. The instrument is once again small enough to fit inside a biological safety cabinet for the dispensing in a nice clean environment. So here's an example of single spheroid dispensing and assays. The image on the left shows a 130 micron spheroid inside the pipette tip. Our collaborator, Mr. Steve McClellan from the Mitchell Cancer Institute, has carried out a cancer stem cell 3D spheroid assay. Single spheroids were plated on the on-chip SPIS, and dose response was monitored over 5 days. Systems currently on the market cannot handle large particles like spheroids, and so our single cell dispenser is indeed a great tool for carrying out assays such as this. So moving on from our sorting technology into our 3D live cell imaging technology, I'd like to introduce the collaboration between PHC and Screen Life Sciences, also based out of Japan. Screen Life Sciences 3D imaging technology covers a wide range of applications from 2D in vitro to 3D ex vivo formats, and we offer 3 unique instruments called the Imager Neo, the Imager Duos, and the ISSCR. Screen's equipment allows you to perform high throughput, non-invasive, and a very high-speed imaging with excellent accuracy. This includes 2 and 3D cell-based assays, angiogenesis sprouting and neovasculature, neurite outgrowth quantification, drug efficacy and toxicity, and the proprietary software within Screen Instrument includes software tools adapted for 2D and 3D imaging analysis and a deep learning analysis tool. We'll go into some of these examples of these applications upcoming. First, I'd like to introduce our optical coherence tomography instrument called ISSCR. It's a 3D live cell imager using infrared laser tomography. ISSCR allows for non-invasive analysis of 3D structures including microvessels, tubular structures, tissues, spheroids, and organoids. OCT provides an advantageous platform to visualize the ex vivo dynamics of the 3D structures, and it can be used as an alternative visualization or monitoring method to obtain quantitative information about the morphological changes occurring in 3D cell culture samples. ISSCR OCT technology enables label-free and non-invasive tomographic imaging and monitoring of microvessels, neovasculature formation, to quantify and evaluate morphological changes in a time-lapse manner. The ability to measure true volume in XYZ directions of 3D structures enables true comparison of drug efficacy in preclinical models. It detects necrotic regions and quantifies volume, internal cavity, tubular structures, etc. with really impressive focus. Also, it's a cost-effective supplementary system to any existing imaging platforms out there. This is a bit of a busy schematic here, but a look at the ISSCR optical system. The OCT observation system adopts an inverted microscope, which picks up the reflected light from the sample, the mirror reflected or backscattered. So now I'd like to walk you through a few examples of some ex vivo applications that the ISSCR technology spans. Again, this is a valuable tool for drug efficacy and regenerative medicine applications, observing in real time the dynamic changes in 3D culture, and also for QC and other biological or diagnostic applications, allowing non-invasive label-free observations. Here's a look at stomach organoids derived from iPS cells. This data comes from Kobe University in Japan. From the scanned area shown on the left, you can see the 3D cut view and cross-sectional observations over long periods of time. Of course, this can be applied to similar tissue layering as well. For evaluating drug efficacy models, we can have visualization of morphological changes within tumor spheroids post-drug treatment with a cross-sectional view. Here's some data out of the University of Tokyo in Japan. You can see the three-dimensional view of angiogenic sprouting and the different quantifying measurements that the OCT allows. The cycling image on the right shows a time period from day 0 to day 7, visualizing this angiogenic sprouting. It's very cool and very valuable analysis that this technology allows. Another example for kidney models and evaluating toxicological effects, this data comes from Tottori University in Japan. You can see the morphological changes due to toxic effects are easily captured and quantified on the OCT system. Here's a recent publication using 3D imaging of bovine embryos. Shown here is a bovine 2-cell embryo with binucleoblastomere imaged by this microscope. On the right, you can see the visualization of the nucleus in blue, green, and yellow of the blastomere. The blue and green nucleus are in the blastomere. So to have a look at another of Screen's instrument, the Duos 2, listed here are some key features, including high-speed whole well imaging for 96 well plates within 1 minute, clear image acquisition for well edge, which reduces the effect of a meniscus, stage fixed design for any suspension cultures, multicolor fluorescence imaging. We have a unique Z stacking and focus synthesis, various plugin functions, as well as a deep learning analysis function, and we'll take a closer look at some of these. This is a quick rundown of the Duos 2 cell imager optical system. Objective range of 4x and 10x. We have 5 fluorescent channels, including 3 automatic. We can do a 2.5 minute fluorescent scanning at high speed, or perform high resolution imaging, which takes 35 minutes per well. We support microplates from 6 to 384 wells, as well as 35 to 100 millimeter dishes. Speed is the key within Duos 2, and you can scan 96 wells within 1 minute and obtain measured results and heat map data, imaging of the whole well as shown in the middle there, as well as area quantifications, as shown on the right. The Duos 2 offers the feature of multiplexing, fluorescence imaging and analysis function for 5-channel excitation light, or up to 3 channels for automatic exchange. Here's a closer look at that clear image acquisition at the well edge. This is an intestinal epithelial organoid under bright field on the left. You can see that using the Duos 2, the meniscus effect is reduced as compared to the bright field. Here we'll have a look at some in vitro 3D spheroid and organoid assays for the Duos 2 and show you some examples of this technology. For organoid cultures, we have the ability to do what's called the Z stacking image of the 3D cell within our Duos 2. And this technology gives us the ability to scan the organoid cultures regardless of where they are in the plate. So you can see here by the dotted lines and going in that Z formation, it takes multiple images and allows that 3D imaging of these organoids regardless of where they lie. Here's another look at that Z stacking scanning. As you can see on the left, the multiple images taken in that on that Z axis creates one composite of an omnifocal image, and this is taken within 3D gel cultured organoid assay. Looking at Screen's analytical software, this includes a tool that allows us to do what's called a deep learning analysis. Our scanning software captures the image acquisition. In this case shown here, organoids were scanned as a bright field stacked images, and that stacked image was obtained by changing focus in the Z direction as you look at that dotted blue line, just like we discussed on the previous slide. And the deep learning analysis of this information integrates into a model file, which we can apply to other test data or to unknown images and offer nice segmentation visualization using this deep learning analysis software. Here's a look at a study we did for in vitro neurite outgrowth assays. We're used to quantitate neurite outgrowth from cultured neurons and neuronal cells. The applications include an assessment of neurite outgrowth potential, the identification of molecules that can block or stimulate neurite outgrowth in 2D and 3D cultures, as well as the identification of agents that can overcome neurite outgrowth inhibition by inhibitory. So here are a couple slides that show an example of how we can follow neurite outgrowth over the course of days and eventually do the endpoint measurements looking at different markers defining from the neurite bodies and into the outgrowth. So shown here, day 0, day 18 in the neurosphere, and day 21 into the development of the sympathetic neuron. And lastly, this is a look at immunostaining of iPS cell-derived sympathetic neurons with the different staining agents that are listed above. And for a further in-depth look at this and really all the other data that I've presented today, please feel free to visit our website and download our great tech papers that we have available. Or please, I invite you to have a one-on-one discussion with myself or our team of scientists globally, should you like to learn more information about these screen products. Now I'd like to move on to another Alliance product, which is a great consumable for the formation of 3D spheroids and organoids. This is called PrimeSurface. First, I'd like to do a quick review of 3D spheroids in today's cell biology world. Since the early 1900s, cell culture has been utilized for studying animal models on the cellular level. Traditionally, a single sheet of cells adheres to the plastic or glass surface of cultureware and forms a single layer where some cell-to-cell adhesion and interactions occur with neighboring cells. Although this method has broadened our understanding of many mechanical and physiological cellular mechanisms and pathways, its limitations are clear when you consider the cell's in vivo state. Simply put, cells don't grow in a single sheet. That being said, a lot of cellular interactions that would occur in vivo do not occur in this single layer of cell culture. Not only do cells in this two-dimensional model not demonstrate an accurate in vivo morphology, they do not have an extracellular matrix. Here I'm going to focus on 3D cell culture that is formed through ultra-low attachment methods. This is done by the way of ultra-low attachment plates, often referred to as ULA dishes or plates. And the plates I'm focused on today are the PrimeSurface series of plates. These are plates that are coated with a unique ultra-hydrophilic polymer that is covalently bound to the plate's plastic surface, enabling cells to aggregate without adhering to the cultureware itself. ULA plates, rather than 3D culturing methods, have shown to be very adapted to existing laboratory protocols, and they can also be utilized for imaging in clear plates, such as on our screen system, or for chemoluminescence screening in more the clinical world, and white plates without any of the added transfer steps. Spheroids grown in low adhesion techniques are consistent in their overall size and shape. This is particularly important for obtaining reproducible results because cellular functions within spheroids are strongly linked to their size. Here you can see two different breast cancer cell lines that were grown in the U, M, and V-shaped wells. The steep-shaped bottom supports weak aggregations of certain cancer cells and promotes tighter spheroids. Researchers have found that the V shape works well with cell lines that demonstrate weak aggregations, while other cell shapes work well depending on the cell line and its affinity for aggregation. In applications that require long-term culturing, media aspiration or dispensing has to be done extremely carefully to avoid disturbing the unattached spheroid. In this case, the culturing of spheroids can be made easier using a slit-well design ULA plate. These 96-well plates allow the media to be aspirated in one step. Rather than disrupting spheroids, this decreases the overall pipetting time over 80%, and it minimizes the risk of experimental spheroid damage. Now, when it comes to stem cells, the PrimeSurface plates play an important role in producing uniform spheroids. Stem cells cultured as spheroids are known to be greatly influenced by the size for stem cell differentiation. Here we have some images taken in the prime surface plates of embryonic stem cells forming spheroids. And as you can see, in less than a week, these uniform spheroids were produced using the M-shaped wells. In cancer research, 3D spheroids have helped researchers take a closer look into drug resistance. It is well known that patients exhibiting tumors and receiving chemotherapy often develop resistance to drugs that were once effective. And according to the NIH, this is true for almost 70% of patients who have solid tumors. Using 3D spheroids, researchers have found that tight junction proteins play a role in inhibiting the penetration of anti-cancer drugs. Shown here are images of human lung adenocarcinoma spheroids that are stained for tight junctions. And even when the images are merged, you can see clearly the consistent overlap of the spheroids' tight junctions. In other aspects of cancer research, 3D spheroids can aid in drug discovery of solid tumors by optimizing high-throughput drug screenings. In this image, we have tumor and multicellular tumor spheroid, MCTS, and you can see the similarities in cell differentiation between the two. Both models feature the same differentiation in microenvironments. The idea here is that 3D spheroids can be more predictive of in vivo anti-tumor efficacy than other cellular models. By creating more complex test systems to gain deeper insights into the effects and functions of molecules in a cellular context, we can optimize preclinical and pre-animal selection of the most active molecules and effectors. So now, last but not least, I'd like to move on to the final category of technology for this presentation. I'd like to discuss our PHCbi brand, which was introduced earlier. Specifically, I'd like to take a couple minutes to look at our new line of multi-gas systems and the importance of hypoxia in stem cell and other cultures. This is a quick look at our PHCbi incubator line, and this line ranges from 50-liter benchtop incubators up all the way into a large 850-liter reach-in incubator unit, which is actually not shown here. They come in sizes of 50 liters, 170 liters, 230 liters, as well as multi-gas models, just CO2 gas models, as well as a variety of different decontamination methods, including ultraviolet light, vaporized hydrogen peroxide decontamination systems, high heat decontamination systems, and a lot of other really nice features for a safe and reliable cell culture. And when it comes to incubator equipment, delivering the most in vivo-like environment starts with a few simple but important design points. At PHCbi, the central principle of design is that we have the best cell health in mind. Starting at the top, to provide the most reactive environment, we need to design with the best brains and nerves, meaning the smartest microprocessor and sensors for temperature, gas, and other diagnostics. For example, if we have a door opening event and cells lose their environment, our infrared CO2 sensor, built with a reference sensor for calibration, will instantaneously recognize and recover CO2 environments. Additionally, our direct heat and air jacket temperature system includes independently controlled door and floor heaters. This allows for nice, even distribution of heat parameters and allows to ensure cells within the chamber are subject to the most uniform environment. To obtain reproducible results from cell cultivation, there must be optimal cultivation conditions during the entire bioprocess. We like to imagine that cells think they are in vivo, and so all the parameters must mimic the internal mammalian environment as closely as possible. Each of the parameters I've listed here are critical and must be maintained with uniformity throughout the incubator chamber to achieve an optimal ex vivo environment. Any changes to temperature, gas concentration, or humidity can create changes in the cells, weaker and inconsistent yields, or total loss of cell culture. Today, we'd like to place a special focus on what we call our multi-gas units, which provide low oxygen environments within the incubator. Of course, we all know that oxygen plays a key role in metabolic and energy-driving cell functions involving ATP. But when you look at the partial pressure of oxygen within the body versus the ambient air that we all breathe, which is generally around 20–21% oxygen, the pressure of inspired air progressively decreases as it enters the lung and travels within the blood throughout the body. So by the time it reaches our organs, oxygen pressures have dropped to roughly 2% to 9% depending on the organ. So the majority of cells internally do not experience the 20 to 21% oxygen that we often culture cells at. Throughout the years of adaptation to using physiological oxygen in CO2 incubators, we've seen certain groups of scientists adopt this method, and subsequently other fields follow suit once they realize the benefit. In the past, it was conventional wisdom to think that the more sensitive or delicate the cell or tissue type, the more prone to physical oxidative cellular damaging that will occur, or that certain cell types are maybe more prone to damages of ambient oxygen. While this might be true, more recent data suggests that hypoxic levels have a much more dynamic role than previously thought. One pioneer is in the PhysOX world is the IVF, or in vitro fertilization industry. Arguably, these folks are dealing with the most sensitive and vulnerable type of culturing work, and thus demand optimized in vitro environment for embryo cultivation. Numerous studies have demonstrated that embryos cultured at elevated O2 concentration levels are impacted negatively during their development, whereas culturing with low oxygen, around 5% levels, results in improved embryo quality, improved implantation rates, and better pregnancy success than at the 20% atmospheric oxygen culturing. This leads to the discussion of the HIF class of proteins. HIF, which stands for hypoxic inducible factors, are a class of DNA binding proteins that become activated at physiological or hypoxic oxygen levels. In virtually any publication that examines PhysOX methods, the role that HIF plays within the biological system will be covered. The HIF protein family is an example of how oxygen plays more of a role than just a quote-unquote damaging agent, but rather acts as a molecular signal for a variety of key cellular events. HIF regulates the transcription of a variety of genes critical for maintaining overall cellular homeostasis in key pathways, such as angiogenesis and glycolysis. So you can see that HIFs play a crucial role in embryogenesis, stem cell differentiation, and different pathophysiological events. Again, more evidence why these fields have adopted this. But this list of housekeeping functions that these proteins play a role in is evidence that really any system or cell being studied should be considered for hypoxic condition. Otherwise, you run the risk that your cells are acting outside of their natural in vivo characteristics, potentially threatening data or downstream events. So, getting back into the industrial trends within the adaptation of PhysOX or hypoxia-type tissue culturing, we're seeing a very strong market adoption of hypoxia cell culturing within the stem cell research and regenerative medicine fields, and for specific methodologies like primary cell culturing and embryo studies. As we've made the case earlier, the trends are pointing towards the wider adoption of hypoxia within a more general cell and tissue culture model for immortalized cell lines, disease models, and to better mimic these disease states under hypoxia. So we can take a look at how PHCbi equipment is designed for experimentation under physiological oxygen conditions. As technology evolved, so did the methods for reading and controlling oxygen in TriGas Incubator. The general principle of zirconia oxygen sensors is that within the incubator, the O2 molecules diffuse through the zirconia layer in the sensor, causing a voltage buildup. The voltage then creates an electrical current flow, which is detected by the sensing circuit within the incubator. The zirconia electrolyte disc has electrodes on both sides, which have the ability to pass O2 ions at a high temperature. This induces a linear current within the electrical circuit that corresponds to the O2 levels within the chamber environment. PHCbi's solid zirconia O2 sensor maintains a long-term, high accuracy while offering a long lifespan. It also does not require periodic calibration, allowing long-term, easy use solution for cell culturing at those hypoxia conditions. So, over the past decade or so, more and more evidence and data has surfaced that points to the importance of controlling oxygen at physiological levels in order to obtain the truest or most in vivo-like data, which is of grave importance to downstream experiments, clinical processes, or simply just experimental conclusions. So, as industries like embryo studies have great success in hypoxia studies, others in the cancer research, stem cell, and regenerative medicine fields have followed suit. Though these cultural methods are still the minority across mammalian-related tissue cultures, we think that more cell culture trends will be moving towards incorporating oxygen into their cell line investigation. And more scientists are examining options associated with conventional CO2 incubators that extend to O2 control as well, giving a lot more experimental flexibility. So a final point I'd like to make, especially to a stem cell audience, is regarding equipment manufactured to GMP specifications. PHCbi brand products have been carefully designed, engineered, and constructed to fit seamlessly into any space or protocol within the clean room and GMP facilities. To ensure that PHCbi equipment can be relied on as an essential tool for cleanroom applications, we've invested in the certification of our flagship cold storage and cell culture incubators to the ISO class 5.0 to 6.0 spec. PHCbi MCO incubators have minimal moving parts and optimize decontamination protocols while minimizing contamination risk. Likewise, our cold storage and ultra-low temperature equipment also engineered to have exceptional reliability, uniformity, and overall performance for trusted short and long-term storage of valuable biologicals has met ISO 6.0. So to wrap things up and to tie everything together, with the expansion of our life science technology offerings, one theme remains constant. All our equipment offerings are designed with cutting-edge Japanese engineering and have evolved with cellular health in mind. This central principle of design holds true regardless of where you are in the research or scale-up continuum. The studies and data discussed today all use the PHCbi family equipment: life cell imaging, cell sorters, cell culture plates, and CO2 incubators. All are built with a unique design and control systems to deliver very stable, very healthy culture conditions and cutting-edge imaging systems. Please, let's have a personal discussion should you like to know how any of these equipment will support your research and your workflows. We have a great team of scientists and experts across the world that will support your work as we continue to evolve our technology systems in the future. Thank you very much. So right now, I'd like to take some time for question and answer. And again, I'd like to thank you for joining us today. I'm happy to take any questions now at any point or offline. I'm showing my contact information here. I hope you all have a great rest of the day. Thank you.
At PHC Corporation, we use the PHCbi brand. If you see laboratory equipment with Sanyo or Panasonic brand, those are part of our proud 53-year legacy of products. Our PHC company is motivated to stay on the cutting edge in new product development, both organically and through strategic partnerships globally. Today, we will share information on our technologies as they pertain to advancements in cell culture, specifically surrounding 3D spheroid culture, cultivation, and analysis. Most recently, we've extended our technology solutions portfolio with the addition of a unique microfluidic cell sorter through our partnership with On-Chip Biotechnologies.